Okay, so when we think about photosynthesis, we have to think about a process that's opposite that of cellular respiration. And if we think about what we did in cellular respiration, we took glucose, right? And we took this glucose molecule and we added it with oxygen in the overall reaction, and we made CO2 plus water, and we made about 36 ATPs. Okay? So that's what we made. That's gas. And that's overall, of course, and this is an aqueous solution usually, but let's just go ahead and leave it like that as a general concept. All right, so then if we're going to reverse this, well, let's think about one more thing about this. Well, the last step, if we broke it down into the three steps that we talked about with cellular respiration, is we took the three steps, which was glycolysis. And we know that in glycolysis, what we did is we took glucose and we changed it into a 3-carbon sugar known as pyruvate. And then from there, it went to the next step, and the next step was the citric acid cycle, right? And in the citric acid cycle, what did we do with that pyruvate? We made 3 ATPs. Three CO2s. We made some ATP. I believe about two ATPs, and we ended up with some NADH, right? And some FADH2. So these were electron carriers. Electron carriers here, both of these. This was used in the process or sent off to do some work. And these 3 CO2s for each pyruvate, right? These 3 CO2s, and ultimately it'll be 6 CO2s because it'll be for each pyruvate it's going to be 3, but there were 2 pyruvates over here, right? So that's what was going on so far. And then in our last step was of course electron transport chain. So if we think about these steps, and they happen in this order, in the first step we broke it apart, right? So in glycolysis, we took sugar and we broke it up into 2,3 pyruvate molecules, and we got some ATP out of it, and we got some electrons and electron carriers, and the electrons went to the citric acid cycle along with the pyruvate. By the way, it's citric acid cycle, Krebs cycle, or tricarboxylic acid cycle. Either way, any one of these ways, there you may see any of these. The key is that in these also, then the pyruvates, as they went on to the citric acid cycle, then got broken down even further. And here at this stage, you produce CO2, right? You produce CO2 gas. You had some NADH. You had some FADH2, right? And up here, you took and you produced pyruvate, right? And you made some NADH. All the NADH went to the electron transport chain. All the FADH2 went to the electron transport chain. But the key is here that all this is going on, and in the electron transport chain, remember that all the electrons got transported, the hydrogen ion concentration was created, ATP is what you made. ATP is what you made. And you had oxygen come in, oxygen gas, and you had it take the last 2 electrons along with some hydrogen ions, 4 hydrogen ions, and you made 2 water molecules, and you have all kinds of animations and videos that showed you all this, and you went over and you did some practice questions, and we went through it like 4 times, and I sent you videos on it, and all that was done, and you should have that pretty much summarized in your head of what's happening here. And there's every kinds of steps, and there's all kinds of enzymes, and all the enzymes have control points, and we're all good with that. Now, photosynthesis is the opposite of this. Let's take a look at what I mean by opposite and exactly what that looks like. So before I do show it to you, let's think about this. What would that mean? What would you expect? What would you think if this was the last step? If this was the last step of cellular respiration, and photosynthesis is the opposite, right? So cellular respiration is glucose plus oxygen equals CO2 and water and ATP. If the opposite is going to be happening in photosynthesis, then you'd expect ATP being put in, water probably being broken up first, and then oxygen into oxygen gas, hydrogen ions then being used somehow, and finally CO2 being put together, and the last step would be CO2 being put together into a sugar molecule, into glucose. So if these were the steps 1, 2, 3, you would think that we would be in the opposite direction in photosynthesis 3, 2, 1. We'd go through an electron transport chain starting with water and breaking it up, going through the electrons being transported, making some ATP, going into some kind of cycle that somehow produces glucose for us or some kind of sugar. Well, that's exactly what happens with photosynthesis, right? We're just taking this and running it backwards. Of course, we need different structures, different enzymes, and we need an energy source, because our energy source for cellular respiration was glucose, but what's our energy source for photosynthesis? I'll give you a hint. It's in the name, photo, and then synthesis. So this is what it looks like. The sun comes in, it leads out its energy. The sun is the source of the energy, hence the word photo. Photons are the source. It's red and blue. Something that's important for you to note is that all photosynthetic materials are green. What are they? Why are they green? What's the only color that they don't use? The only color that they don't use. Well, think about it, and I'll try to tell you in class, but think about it. You should be able to figure it out. I hope you see that they use the red and the blue, and I'll give you this much. Remember, the colors of visible light, they're going to use visible light, and it's red, orange, yellow, green, blue, indigo, and violet. And according to this, you know they use the blue at least, and the red, because you see the light there is blue and red. I'll give you another hint. They use indigo and violet somewhat, to some extent, and orange and yellow less so. But so what color don't they use? Remember, all photosynthetic plants are the color blue, green. They're green. So think about what's the only color that they don't use. So in any case, here's the light coming into the plant. We have red and blue. It excites the electrons in water in the photosynthetic piece, and we'll look at what that looks like in a sec. And then the very first thing, as I said, you're going to take water, you're going to break it up, you're going to release oxygen, and then those hydrogen ions are going to be used in an electron transport chain. And here it is up here, the light-dependent reactions. You have 2 photosystems, photosystem 2 and then photosystem 1. And in both cases, what you're doing is you're exciting the electron that runs it through an electron transport chain that pulls the hydrogen into a higher concentration. And this is what it looks like, the hydrogen ion concentrations going up into the high, right? So you're making the high up here and the low on the other side. So this internal section of the thylakoid, this is known as a thylakoid. These little discs, there's many of them, to try to capture as much light as you can. In photosystem 2, the very first thing you do is you start to fill the space with hydrogen ions. And of course, we have our friend ATP synthase. As hydrogen ions are inside, when they're high, and this out here is low, in the stroma, the very first thing that happens is they go from high to low, and they make hydrogen ions, and they make ATP. The ATP is going to go into something else, though, and we'll talk about that in a minute, but we're making ATP from the sun. We got the energy sources first in the sun, and now we're making the ATP. So, and you can see it here. Here you have a high concentration of hydrogen ions. How do we make those hydrogen ions high concentration? We use the sun's rays to break up the water into hydrogen ions and electrons, and those electrons get carried through carrier proteins, just like we saw in the electron transport chain in respiration. And the high concentration of hydrogen ions escaped through the ATP synthase, and ADP went to ATP. You have a lot of ATP. Well, that's the key. That's it. There you go. You have a lot of ATP. We captured the sun's energy. We made ATP, but that's not enough, is it? Because the plants need sugar. That's the point, is to get sugar, not to make ATP. Making ATP is nice, but ATP only lasts a short term. We need a little bit longer term storage of this energy. We need to put this energy into a form that we could use, right? That plants can use. Sugar is a great source because plants have mitochondria, and so if you have mitochondria, you can do cellular respiration. But what do you need to do cellular respiration? Sugar. And plants are made of wood, and we know that wood is made of cellulose, and cellulose is made of sugar, glucose. So glucose is important. Sugars are important, and so they need to be made. These carbohydrates have to be made. Well, we produce the ATP. That's the energy we're going to put into the sugar. How do we do that? Well, in the photosystem 2 and then photosystem 1, the whole objective is to get the electron carriers, NADPH. Now notice it's NADPH, a little different from NADH, but don't worry about the difference. It's almost not, it's not, at this stage, it's not even an issue that we should be talking about, but it's NADPH, and I know some of you are going to notice it, so I'll point it out. It's just slightly different. And the NADPH, electrons for NADPH, and ATP that we produce here with these electron transport chains, oxygen's already left. The water's broken up. We're going to take the NADPH, the hydrogen ions, and the electrons and ATP. We're moving it into what are called the light-independent reactions. And that's a cycle. Now remember, I'm going to just remind you for a second what I said in the beginning of this presentation. The first thing is everything's in reverse, right? So we started off with getting energy. We got it from the sun. Then we put it into the electron transport chain here in photosystem 2 and then photosystem 1. They're just two different photosystems. They're used to capture the light's energy, the sun's energy, and create this high to low concentration of ions to excite the electrons and make sure that they're excited. We have the oxygen's gone, right? We have the ATP, we have the NADPH. Everybody's happy, we're ready to go. Where's it going to go? These are the light-dependent reactions. They're dependent on light. These used to be called the dark reactions, but really they happen in the day or night. They're just independent of light. They don't have to happen with sun present, the light present. These reactions do. In order to have the high concentration inside, low outside, you have to have the sun present. You need to have water, you need to have the sun. That's why plants need water and the sun, because they have to break up that water and use those electrons. So now they have them, they need to move these resources over to the light-independent reactions. And the light-independent reactions are pretty straightforward. They have to capture CO2, because what is glucose made of? What did we produce when we broke up glucose? We produced CO2. So it makes sense that we're going to then, if we're doing the opposite, we're going to take CO2 and we're going to put it back into glucose. Okay? We're going to take CO2 and we're going to make glucose out of it. So CO2 comes in, it's captured by a really interesting enzyme, probably the most important enzyme on the entire planet. That's a pretty big claim, but I'll go ahead and say it. The most important enzyme in the whole planet is Rubisco. And there it is, RUBP. Just call it Rubisco. Rubisco's whole job is to capture CO2. This one enzyme probably changed the atmosphere of the planet. This one enzyme allowed animals and plants to coexist and transform the entire world. So probably the most important enzyme on the whole planet. And there it is, rubisco. And it captures the CO2. It takes that CO2, and that CO2 enters the Calvin cycle. Now, this is not the Krebs cycle, right? In the Krebs cycle, what did the Krebs cycle do? It broke the pyruvate into CO2. It actually made CO2. The Calvin cycle is the opposite. Everything's opposite here, right? The Calvin cycle is taking the CO2 and making the sugar. Where the Krebs cycle, you made the CO2, the Calvin cycle captures the CO2. So they're just opposites of one another. As you make the sugar, of course you use the electrons that, that in cellular respiration you rip off the sugar. Here you put the electrons back in with the high energy electrons. You're producing hydrogen ions. Of course, the hydrogen ions can go right back into the system. And you use that energy in ATP to make that sugar. You're putting that energy into the sugar by putting into the bonds between the carbons that made that 6 carbon side hexagonal shaped molecule, right? That sugar. And that is, and in fact, it should say we make a 3 carbon sugar known as PGA. There's well, let's just not, I don't even want to say it. It's going to upset some of you. It's a 3 carbon sugar that then get 3 carbon sugars, 2 of them get together and they make a glucose. But the point is that this is the process, this is the general process of photosynthesis that you need to know. All right, I tried to use as few words as possible and try to keep it as simple as possible. And just to say the respiration, you take sugar. In glycolysis, you split it up into 3 carbon sugars. That's the first step, right? You take some of that ATP and electrons. The electrons you send through electron carriers, the electron transport chain. The ATP, use it as you will. The pyruvate goes into the mitochondria, gets broken up in the TCA cycle or citric acid cycle or Krebs cycle into CO2. More electrons are put in electron carriers. They're set in an electron transport chain. You create a hydrogen ion concentration. That chemiosmotic gradient allows for the potential to be there to create ATP. You allow ATP synthase, make that ATP by allowing the hydrogen ions to flow through it. And the oxygen is the final electron acceptor, taking hydrogen ions, oxygen, and the electrons and making water. Well, again, the opposite here, photosynthesis. The very first step is the last step of cellular respiration, and you're taking water and you're splitting it up. Taking water and you're splitting it up into oxygen. The hydrogen ions go into the system. The light excited them. ATP gets sent over to the light-independent reactions. The Calvin cycle, which captures CO2. The CO2 gets put into sugars. The hydrogen ions get kicked out. Electrons are all in the sugar. The energy's in the sugar. And so now that sugar can go and be used in cellular respiration, or be used as a structural component of cellulose, or be eaten by a cow and then turned into other things, and whatever else is going to happen. So this whole process is a giant cycle, which goes back to that picture that we started cellular respiration that you learned in middle school, where you saw the plants breathing out oxygen. There it is. And the animals taking in oxygen. I want to point out again that plants do have mitochondria. Plants do use the oxygen that they produce to break down the sugars for energy when they need to. Too many people in the world think that plants don't do that, just because you were taught that plants take in carbon dioxide and put out oxygen, which is true. As you see right here, it's true. They take in carbon dioxide, they put out oxygen. The chloroplasts do. But they also use that oxygen, and they use that sugar that they produce, that they took and made the CO2 into this sugar. They take that sugar and they can use it for energy in cellular respiration in the mitochondria and in the cytoplasm. So it's really pretty important you remember that. I think it's a little important that you understand what some of this stuff, what's going on in photosystem 2 and photosystem 1. I'm not quite sure if they're going to ask you a question this deep, but I think it's important that you understand what we're dealing with here. You do not have to know these structures, so before you go freaking out that, oh my God, do I have to memorize the structure of these proteins? And absolutely not. I just want you to see that they are proteins. They're big, complicated things. That chlorophyll, there's the pigment sitting there in the membrane, is one of the first things it does is it takes this water, right, as it captures the light, and the first thing it does is take the water and then break it up into oxygen gas. All right, so Ru, right, this complex, helps the water break down into oxygen gas and hydrogen ions, and there it is, right? And this electrons that got ripped off of water get sent down the electron transport chain. That's important that you understand that. As this is photosystem 1, photosystem 2, the whole job of photosystem is to take that light and rip off the electrons, and the electrons are there. Photosystem 1, its job is to take some more light and put it into the electron so that it makes it a high energy electron. And what that's going to do is it's going to put that electron into NADPH. Then that electron can then go on to the into the light independent reactions, right? And in light independent reactions, we can take and then combine them. And of course, when you do that, when it goes off in there, one of the things it does is that you've already created over here hydrogen ion concentration. You see that here, right? That this electron coming in here, you saw the hydrogen go from here where it's low to here where it's high, and it zooms in here and makes it high. And of course, over here, this ATP synthase takes that hydrogen ion and lets it flow through and make ATP. Then ATP and NADPH can come over here to the light independent reactions, which is badly drawn, but it's just a really good overall picture. You see that the CO2 is captured by Rubisco, then it's taken in, ATP is used, the electrons are put in, and you make the sugar that you're trying to make. So for most of the history of the world did not have a lot of oxygen. And as such, Rubisco was a great, evolved in a world without a lot of oxygen. And so CO2 was present, and Rubisco worked really well. But as oxygen became more plentiful, and especially during times of a lot of heat, with oxygen being present, Rubisco does something that is unfortunate for the plant and for the rest of us. Rubisco chooses then to bond with the oxygen and then makes this, ends up making CO2, oddly, and using ATP and NADPH. So it actually does photorespiration instead, and this is why it's called photorespiration, because it's producing CO2 and using ATP. And then ATP gets used up again up here as it goes back to Rubisco and makes another, and takes another oxygen and continues the cycle, wasting the energy that it's taking up. This happens in every plant all the time, but it's not a big problem as long as the heat, as long as it's not too hot, because the Calvin cycle is still rolling and this doesn't happen as often. But as the temperature increases, like in the tropics or in the desert, it becomes a problem because oxygen is present and that's an issue and it's hot. So plants have evolved in these places, in the tropical areas, and in the desert, it's handled. It does two different kinds of photosynthesis. The first is C4 photosynthesis, and the next one is a CAM photosynthesis. And in the C4 photosynthesis, what it's doing is it's taking the CO2 from the air, and as it comes into the cell, it binds the CO4, the carbon dioxide, using this enzyme called PEP carboxylase into a 4-carbon compound. And then it transfers, it separates photosynthesis into two parts. first the part of capturing the CO2, and that's done up here, all right, in the outer cells where it's exposed to oxygen. But then it takes and transfers that CO2, that carbon, in with this C4 compound, a 4-carbon compound, into this cell. And in the inner cell, there's not much oxygen at all, or not a lot of oxygen, as compared to the cell on the outside that's exposed to oxygen. And it releases that CO2 and then continues on back around its own cycle, the C4 cycle. So it goes up, picks up the carbon dioxide, becomes a 4-carbon compound, releases carbon dioxide, goes back and becomes a 3-carbon compound, and keeps going round and round, picking up a CO2 every time. And it dumps the CO2 in the inner cell where there's not a lot of oxygen at all. And here Rubisco works fine. And you don't get a lot of photorespiration here because there's not a lot of oxygen here, even though it's hot and there's a lot of water. It's in the tropical areas. So over here is tropical, right? So we're talking about things like, pineapples and such, where they grow up in the tropical areas. They're going to want to do this kind of photosynthesis because it's going to be a way it's going to be able to do its capture. Rubisco is going to be able to be used in a place where there's low oxygen levels. Even though it's hot, it's hard for photorespiration to happen when there's not a lot of oxygen around. Now, in places where it's really hot and dry, like in a desert, so things like cacti and etc., they're going to want to separate the photosynthesis process by time, as well as trying to reduce photorespiration. It wants to reduce the water loss. So there are these openings called stroma, not stomata, but stoma. So it's not stroma. Stroma is the part of the chloroplast, is a space inside the chloroplast. It's stomata, and in the stomata, what you have is an opening that is used by two different guard cells, or is protected by two different guard cells. And I know it looks freaky, but that's what's on each leaf. When you eat a leaf of lettuce or spinach or any kind of leaf, you have these openings in here, and these guard cells, there's a cell on this side and this side, they swell up and they close, or they shrink and they open up, and that allows gases in and out. So they look like mouths, and in fact, they act like nostrils as well. They let air in, but unfortunately they also let water out, and desert plants don't want to let water out, so they open these stomata at night. So the stomata are open at night, that lets CO2 in, oxygen out. PEP, carboxylase, captures that carbon dioxide and turns it into a C4 compound, right? Then the sun comes up, the stomata close. So now there's not a lot of oxygen in here because the stomata are closed. It's hot, so they can't lose their water either. And that 4-carbon acid releases its extra carbon to become CO2. Then it becomes a 3-carbon acid, which goes back into PEP and starts to cycle all over again. Meanwhile, the CO2 can enter the Calvin cycle, and Rubisco can capture that CO2, high levels of CO2, low levels of oxygen. Even though it's hot and dry, the water's not being lost because it's not open during the day. And that CO2 is captured and put into glucose. And the plant wins. So in both the tropical and in the desert areas, in the hot dry and the hot wet, you have two very different evolutionary advantages to having these two different kinds of photosynthesis. In places like Cleveland, where it's not very that hot, comparatively not hot, where it's wet enough and cool enough that photorespiration isn't an issue, these photosynthesis don't really happen unless you have a tropical plant that you happen to be growing here. In the tropical regions, you definitely want to have this separation of processes in space so Rubisco doesn't get confused and do this awful thing called photorespiration. And in the hot, dry areas, the plants want to save their water and close their openings, their stomata. And here, this is what they look like. Close the stomata at night, during the day, and open them at night, letting CO2 in, capturing the CO2. Remember, the whole point is to capture the CO2, capture the CO2. In the regular Calvin cycle, that would in plants that would happen in the day, but in places where it's hot and dry, we don't want to capture that CO2 in the day. We would want to do it at night. And in tropical areas where there's a lot of possibility, a high degree of probability that there's going to be photorespiration occurring, you want to separate it by space as well. So this is key, that you think about the evolution, that plants have evolved to their different environmental to their different environments to survive, and that these enzymes evolved a long time ago. They're very conserved. They said the most important, probably one of the most important enzyme in the whole planet, this rubisco. And it evolved a long time ago when conditions were very different. And plants have had to evolve and change. And and this enzyme is so important that it's been kept over the millennia, over the eons. It's been kept, and whole structures have evolved to try to compensate, right, for these environmental changes. So we're in hot, dry areas, you have stomata that open and close in different times, and you have this whole pep carboxylase, an extra enzyme that evolved to capture this thing. And over here you have a whole development, a whole tissue, an entire tissue of mesophyll cells whose whole purpose is to capture CO2 and transfer that CO2 to the bundle sheath cells, where they can conduct the making of sugar without the bothersome photorespiration that wastes the energy. Okay, so that's it. That's photosynthesis. Again, it's the opposite of cellular respiration. I hope I was clear. It's a little late at night, so I will clarify anything that needs clarification on Monday. I'll see you on Saturday school. All right, have a good one.